So in this experiment, the MSH2 gene that is linked to to HNPCC, a cancer diagnosed in humans, was studied. The MSH2 gene is a DNA mismatch repair gene responsible for HNPCC. A mutation in the mismatch repair gene increases mutation rate. The higher the mutation rate, the greater the chance an individual has of being diagnosed with HNPCC. The mutation in the gene G6, G693D was studied using budding yeast. The purpose of this experiment is to examine the plasmid, or the wild type mutant and empty vector carrying the MSH2 insertion, and determine the pathogenic nature of the mutation. Results show that there are significantly less colonies are shown in the MSH2 section than the empty vector. However, the mutant yeast had even less colonies than the wild type. Although the mutant is benign, it should not have a better DNA repair function than the wild type. The transformation of the yeast with PSH44 plasmid and the plasmids containing MSH2 alleles was successfully, as indicated by growth of yeast colonies on the selective SD his TRP plates. HNPCC is a condition driven by genetics because the probability of contracting colorectal cancer is inherited and increased. MSH2 was the first gene linked to HNPCC to be sequenced. From this sequencing project, researchers concluded that other MMR genes were involved. HNPCC is caused by a mutation in either HMSH2, HPMS1, MSH6, chromosome 2, MHLH1, chromosome 3, and HPMS2, chromosome 7. And figure 1 is a display of this. Budding yeast. Yeast are used because trials on humans are not legal, and understanding the role of MSH2 in yeast will help us better understand the role of MSH2 in HNPCC. So the Bradford assay. Bradford protein assay is an analytic procedure used to measure the concentration of protein in a solution. The purpose of this lab was to find the concentration of an unknown protein. The materials that we used in this lab were 5 times Bradford assay reagent, 100 micrograms per milliliter of bovine serum, or BSA, 0.9% of NaCl solution, protein sample with an unknown concentration. So our methods consisted of 6 standard reactions containing various volumes of BSA stock solution and protein concentrations that we set up. Then the 0.9% solution of NaCl was added to each reaction in order to reach the final volume of 1 microliter in each reaction. As we sampled with a one-time Bradford reagent and 50 microliters of an unknown protein concentration sample, we also set up. So the NaCl was added to the reaction until the final volume was 1 milliliter, as stated before. All the reactions were labeled according to their protein concentration. For instance, unknown, STD1, STD2, etc., and mixed thoroughly. The reactions were incubated at room temperature for approximately 5 minutes. Each reaction was measured on a spectrophometer at 595 nanometers absorption. The absorption of all the samples were recorded. The standard curve was plotted using Excel, and the concentration of the unknown was calculated. And here we have a visual interpretation of this in our figure. So figure 1 is the table that shows a breakdown of the measurements of each component that was put into each reaction tube. And then figure 2 is the table that shows the absorption of each reaction measured using the spectrophotometer. So the second experiment with isolation consisted of materials that were DNA samples, lysis buffers, neutralization buffers, an endo wash buffer, zippy wash buffer, zippy elution buffer, and the methodology consisted of spinning reagents and I'm off, sorry. The methodology consisted of centrifuging the bacteria culture. The DNA was located in the pellet. The super, all the excess was discarded. Using 600 microliters TE, the bacteria samples were resuspended. 100 microliters of the lysis buffer and 350 microliters of the neutralization buffer were added and inverted 2 to 3 times. The substance was centrifuged for about 4 to 6 minutes. The DNA was the DNA was in the excess and transferred to new collection tubes from that centrifuge for about 15 seconds. Flow through was discarded. 200 microliters of endo wash buffer and 400 microliters of the zippy wash buffer were added, then centrifuged for 1 minute. 30 microliters of the zippy elution buffer were added to the solution and poured into a new tube. Each sample was centrifuged and the purity and concentration were recorded. And below in table 1, you can see all the results that were yielded. Um,
So the next project is the materials and methods that we use for the restriction enzyme analysis. We used a double distilled of water, and it was labeled as DDH2O, and we use SP0, SP1, and SP6, and we use a digest buffer and X-ball of 5% and A-ball of 5%. The methods, once we started the experiment, we had to spin the reagents after mixing all of the mutation buffers, and then we diluted each plasmid DNA sample with double distilled water, which is it was labeled DDH2O. One set of samples contains one fast digest buffer of the 5% of the X-ball and the 5% of A-ball and the 500 nanometers of the plasmid DNA. And we used a second set of samples that served as a control reagents that were actually undigested, containing one fast digested buffer and 500 nanograms plasmid DNA. And we incubated the reaction at 37 degrees Celsius for 30 minutes, and then we prepared agarose gel in a TAE buffer and assembled the gel in the tray and poured the agarose gel into the tray until it became more solid. Then we had to perform a gel electrophoresis on the following samples. And we got to make a DNA ladder with the 3 mutations that is listed above. We had 10 milliliter digested products, which is 3 lanes, if you see to the right here. And then we had 10 milliliters undigested products, which is 3 lanes. So we had a total of 6. So after the electrophoresis images, we imaged the gel, which is to the right. Should be right. So we also use, excuse me, a second. So we also used the materials for the polymerase chain reaction. This was to amplify the region which contained the mutation. And the materials that were used are listed right here. It was all given to us by Dr. Chen and Dr. Kyoko in the lab, which was the ready-to-go beads in the 0.2 milliliters of the PCR2, the diluted DNA, and that's the SP0, SP1, and the SP7, and a nucleus-free water, the solution containing 10 micrometers for primer and reverse primer. First, we added the 10, this is the figure right here that's explaining the polymerase chain reaction. This is what we had to actually digest to understand the lab and what we were doing before we even started the lab. But the methods, we used 10 nanograms plasmid DNA and then 0.8 microliters primer and then a bead. Then we flicked to mix, and all the tubes were placed in the thermocycler for the program. The next was the restricted enzyme digestion. So on the PCR fragment to see which one contained the mutation, the agarose gel was used to see how the restriction enzyme digests the wild type and the mutant. So we used the double distilled water as we did in 2 labs ago that I explained, and the restriction enzyme, which was the BSMA1 and the buffer and the loading dye. So we diluted the buffer with the 100 to 200, and then we digested 3 tubes of the buffer and added the 2% of the BSMA1 and 10 microliters of the PCR product. Then we got another 3 tubes, which were labeled undigested, and we used the buffer and just 10 microliters of the PCR product. We incubated that at 55 Celsius for 1 hour. So next, this is our result. We had the transformation. This was the process that placed the plasma into the yeast cells, and we got a plate, and we had to label our cells into different cells, and it was labeled according to what Dr. Kiyoko and Dr. Chen provided us. And we used reported plasmid. They used microsatellite, and it provided functional analysis of the MSH2 mutation rates at the different concentrations. So after we got done with that experiment, were able to create the flow cytometry analysis in our studio. So first, this is a technology that is used to analyze the physical and chemical characteristics of a particle in a fluid as it passes through at least one of the cell components, and they're labeled and excited through a laser by light and varying wavelengths. So the fluorescence can be measured to determine the various properties of like each single particle. So which are usually cells, and we refer to them as cells instead of particles. And up to 1,000 particles per cell can be analyzed as they pass through the liquid stream. And this is just an example that we did in class, and this was also provided to us from Dr. Chen and Dr. Kiyoko. We used the optical to electric coupling system, and this is the research that we did on this process. We used a optical to electric coupling system is used to record the way it is which the particle emits fluorescent and scatters indents and light in from a laser. It was 3 main streams to make up for the flow symmetry instrument, and these are fluidics, the optics, and electronics. The purpose of this system is to transport the cells or slash particles in a stream of fluid so we can be able to interpret what we need to know and also to integrate. So each one of these sections right here is explaining the different phases of cell as in G1, G2, and all that to G0 in a way. So we use this to analyze that in the experiment. And this is the graph that we came up with. So as you can see, the VEC-HU and the NLPL is not in the same category, and the VEC-HU is also hindering over to this way. So we were able to differentiate and talk about in class of why this was the way it was. So our overall conclusion came to that the objective of the MSH2 project was to identify the pathogenic nature between the MSH2 strain and the 3 different plasmid DNA samples based on fluctuality. It was determined the mutation occurred was G693D was the binding in nature. So first we have to explain lastly we have to explain our experimental errors. So a couple of the experiments we had to predict or analyze what we would think would happen because my partner and I were either absent due to research or our GRE standardized testing, and we are not able to complete the experiment. But we came up with different errors. So some of our research project was done by educated guesses. So we did pipetting errors caused the original DNA sample to be too small. So that was probably a problem with the improper usage of the pipetting. Centrifuge problems, and with the gel electrophoresis external puncture of gel, the wells were towards the positive electrode and had to be rotated. Additional too much temper to the final restriction enzyme digestion. It did not have a major effect on the gel picture. These are references that we did that are proper research on the project before, and that's it.